Hi, this is Dr. Bueller, and we're going to talk about Alzheimer's disease and the pharmacology of anti-Alzheimer's agents. When we think about Alzheimer's, what we normally think about are learning and memory deficits and cognitive deficits. And while it is a lot more complicated than that, we can really kind of bring it down in a nutshell to the fact that those deficits are caused by degeneration of cholinergic neurons in the memory and cognitive pathways. We consider Alzheimer's disease to be a neurodegenerative disorder. You're born with this function and learning, memory, and cognitive functions working fine. But if those neurons start to degenerate, then you're going to lose function in those. Obviously, our goal with any neurodegenerative disorder is to stop neurodegeneration and hopefully to stop it before it ever becomes a problem. If we can't stop it, we at least want to slow it. And worst comes to worst, if we can't slow it, we at least want to try to boost the function of any neurons that are remaining. When we look at Alzheimer's, we can look at it in the scope of the cognitive, psychiatric, and motor functions that we see in all neuropsych disorders. And while we normally are thinking about those learning and memory and cognitive deficits, we can also see that we have deficits in language use, other types of problem solving, short-term memory, as well as loss of long-term memories, all under this cognitive domain. The disease progresses, and as we look more thoroughly, we can also see some motor deficits, such as ataxia, loss of coordination, and in the worst cases and later states of the disease, things like difficulty swallowing. Psychiatric components include agitation, anxiety, depression, and we're not talking about just as a byproduct of, say, depression because you have this disorder and you're trying to deal with it, but changes in the neurological pathways for anxiety and depression themselves. One of the things we want to do is define the term dementia. Dementia is a very general term. It doesn't just mean loss of learning and memory. It's memory, cognitive function, and personality changes. There are a lot of different ways you can get dementias. Alzheimer's is not the only form of dementia. We're focusing on the pathophysiology for Alzheimer's dementia. We look at Alzheimer's disease. We talked about how this is due to neurodegeneration of cholinergic neurons. As the disease progresses, some non-cholinergic neurons are going to come in as well. But really, this is where all our drugs and all our drug development is focused on. We can have familial, early onset Alzheimer's, and we can have later onset Alzheimer's, which is much more common. The familial or early onset is when it comes before age 65 and is because of a very simple genetic factor. The sporadic is much more common, comes on after 65, and it also has some genetic factors. So we want to be clear that sporadic does still sometimes run in families, but it has to be the genetics plus environmental risk factors, and it's much less predictable. We have progressive symptoms from early to late stage. And unfortunately, at this point, we don't have any way to stop the degeneration from occurring. I want you to take a look at your acetylcholine neurotransmitter notebook page. When we talked about the disease state itself, we're talking about decreases in acetylcholine while those cholinergic neurons are dying in the memory and cognitive pathways. And then when we talk about the drugs, we're going to be talking about drugs that are going to be increasing acetylcholine. And so we're going to have ADEs that are going to match procholinergic effects. So please be sure that you are familiar with your procholinergic or promuscarinic effects. Sometimes you may use an acronym like SLUDGE. It's the one I know, which is salivation, lacrimation, urination, defecation, GI, and emesis. So we consider those the wet symptoms, and we consider here these low cholinergic or anticholinergic to be the dry symptoms. So what's causing neurodegeneration? Well, we know that the neurodegeneration is quite profound and quite pronounced. If you can take a look at this picture of this brain over here, we see that in the Alzheimer's brain, we have significant decrease in the actual volume or size of the brain regions involved in memory, involved in language use, and up here involved in cognition. We actually have enough loss of neurons themselves to see the shrinking of the brain matter this clearly. As the disease progresses, you might see other neuron types and other pathways involved and begin to degenerate as well. But the most susceptible ones and the ones that have to do with the learning, memory, and cognition are going to be, as we said, the acetylcholinergic pathways and the ones in the cognition and language regions. So what happens when these die off? Here's our normal cell or our normal person. And we have two regions we're focusing on, the hippocampus that creates learning and memory, and the cortex where we have cognition created. In both of these regions, I'm just going to show it up here at the top in the hippocampus because the same thing is happening in the cortex. What you should have in the healthy brain are a lot of cholinergic neurons that are coming into the hippocampus, releasing acetylcholine at synapses in the hippocampus, and then activating postsynaptic cells that are going to take that information out. To encode memories, to retain and to retrieve memories, we're going to want this pathway to function properly. We remember that we have nicotinic and muscarinic receptors. And so nicotinic were those lichen-gated ion channels, and muscarinic were those G protein coupled receptors. And we have both of those types of receptors involved in these synapses. And those main cholinergic pathways that we see involved are going to be in the hippocampus and here in the cortex. The disease state is when we start to see that neurodegeneration. So here in the Alzheimer's brain, we can see some of these cholinergic neurons are dying out. And as they die out, we're going to have less acetylcholine being released. So we have fewer inputs into the hippocampus, less acetylcholine being released. We're also going to see death of these postsynaptic neurons. And so we're getting less input, less transmission, and less output. So basically on all of the parts of this synapse that we can mess up, we have loss of presynaptic cholinergic, loss of cholinergic receptors, even in the cells that are still living, and a loss of postsynaptic cells. Luckily, each of these pathways is not just one neuron to one other neuron. That's why I show 3 neurons here going to the same places. We have multiple neurons creating the signals for each form of memory and for different cognitive pathways. So when 1 or 2 neurons start to die off, you can still have enough to keep that pathway going. But as you lose more and more of these neurons, you're getting less and less signal able to go through, and you get less function until you reach the point where you don't have enough signal going through at all to have any physiological function still available. So we said that these neurons are dying. We have some pretty good ideas of why they're dying. We see in Alzheimer's brains that we have 2 major structural changes. We have outside of the cells what we call amyloid plaques, and we have inside of the cells what we call neurofibrillary tangles. So the extracellular plaques are made of amyloid beta 42 protein. We can also call that beta amyloid 42, A beta 42. It all means the same thing. These guys are proteins that are natural in the brain to make amyloid proteins. Amyloid 42 is especially problematic, and what it does is it starts to build up around the neurons, and A beta 42 itself is somewhat neurotoxic. We also see these neurofibrillary tangles. These are made up of tau proteins that are normally functional, but they're hyperphosphorylated and now no longer have function. So let's say it's a little tau protein and it gets hyperphosphorylated. It can't do what it needs to do. What it needs to do is to help the function of microtubule transport. And so if you guys remember, you have the cell body, and then so you go down through the axon to the presynaptic terminal. Microtubules were that function that helps you move proteins, carbohydrates, lipids. And so if you're not moving proteins, enzymes, etc., from the cell body where they're being made properly to the presynaptic terminal, then that neuron's going to die, that presynaptic terminal's not going to work. and you're going to end up with cell death. So you have those A beta 42s that can be neurotoxic and that clump up in those amyloid plaques around the outside of neurons. And you've got these hyperphosphorylated tau proteins that are causing reduced microtubule transport inside the neurons. And all of this is neurotoxic. So we know we've got the amyloid, we know we've got the tau, and we also know that we have a lot of neuroinflammation in these brains. So we're going to talk about each of these hypotheses really briefly, but when you look at a disease state where are these multiple hypotheses out there, it's going to be pretty likely that there's at least a little bit of all of them going on. So I think it will be very unlikely if we don't see some amount of feedback interconnecting all of these hypotheses. So with the amyloid hypothesis, it's that beta amyloid 42 is the cause of everything that goes wrong. That it gets made, it gets clumped up into plaques outside of the neurons, and that the toxicity because of the beta amyloid is what causes the tau proteins inside the neurons to misfold, causes that dysfunction of microtubules, and that these amyloid plaques outside the cell that aren't supposed to be there, that look like trash, are also going to be recruiting immune cells that think that that's an invader or degradatory products that need to be gotten rid of, and those inflammatory cells are being brought in because of the A beta 42. So that idea that the amyloid is really what's causing everything to start. The next one is that tau toxicity hypothesis, and it's just that they believe that the tau phosphorylation is the first thing to start, and that tau phosphorylation that is reducing the microtubule function is causing the neurodegeneration and is also causing the amyloid plaques or the amyloid beta 42 to build up. Also, then that toxicity is what brings in neuroinflammation. And like you guessed, the neuroinflammatory hypothesis is that neuroinflammation is the first thing that's happening outside of these cells, and that all of the chemical mediators and inflammatory cells that are coming in to deal with the inflammation are actually attacking the neurons themselves, and that's what causes the tau hyperphosphorylation and causes the buildup of A beta 42. So all of these things, hypotheses agree that all of these three things are happening. They just disagree on which one starts the whole thing. So I'm going to try to explain to you the amyloid hypothesis, but you do not need to memorize this. So I'm going to explain this slide, and then we're going to go to the next slide, and I'm going to point out what you really need to know. But I want you to have seen the whole picture so that when we get to those little drug targets, they make sense. So we said that we have A beta 42 or beta amyloid 42. That's our main culprit here when we talk about the amyloid hypothesis. The reason we have A beta 42 is it's a breakdown product of what we call APP, amyloid precursor protein. And amyloid precursor protein is good, it's necessary, it's involved in the membranes of neurons. So we're perfectly happy that we have this APP. It's just sitting here in the cell membrane with a lipid component so it can sit in the membrane, and then this protein component on the outside. When that APP gets old and starts to degrade, though, we want to break it down and we want to get rid of it. So there's a couple of different ways to do that. We use a combination of these secretases. We have an alpha secretase, beta, and gamma secretases. And depending on which secretases we use, we're going to clip that APP into two different sizes of amyloid beta. If we use a combination of gamma and alpha secretases, we're going to clip this into a shorter version called A beta 40, which is fine, no problem, we're good. Or we're going to use, on the other hand, a gamma beta secretase combination, and that is going to clip this into a bit longer version of beta amyloid that's 42 amino acids long. And that's where we have our problem. Clipping here at the gamma, clipping here at the beta, and now we end up with this beta amyloid 42. Beta amyloids are normally made by the brain in the breakdown of APP. So this is something that's normally going to happen. And those beta amyloids are going to be taken away by a shuttling molecule called ApoE. So ApoE is also used in the bloodstream and it moves cholesterol around. When it's here in the brain, it's grabbing onto beta amyloid and it's taking it away. This all seems fine, and it normally is as long as you have A beta 40 and you have good functional ApoEs. If you have A beta 40, this binds really well to ApoEs. And so that A beta 40 is just taken away, gotten rid of, and you don't have any buildup and you don't have any problem. However, if you're making a lot of A beta 42, it does not bind well to ApoEs. So what you're going to have is you're just not going to clear out that A beta 42 as well as you would have cleared out A beta 40. So now say we're making a lot more A beta 42 than we really should be, and it's not getting cleared out. We're going to start getting a pileup of it. Here's a single A beta 42. As there gets to be more of it, they start to stick together into protofibrils. And then those protofibrils stick together, making up fibrils. And all those fibrils come together and just start sticking to each other until they form those plaques. Now, I said that A beta 40 is cleared much better by ApoE than A beta 42 is. That doesn't mean that A beta 42 is never cleared. We have different isoforms of ApoE. So we have ApoE 2, 3, and 4, the ones that matter for us here. All of these 2, 3, and 4 are going to shuttle away A beta 40 just fine. But when it comes to A beta 42, they have really different functionality. So if you have ApoE2, that's the best case scenario. ApoE2 is going to bind and clear A beta 42 the best of any of the ApoEs, although it still doesn't clear it as well as it would clear A beta 40. So if you're going to have a lot of A beta 42, you want this ApoE2 isoform. The ApoE4 isoform is the worst one to have. It has the least binding and clearance of A beta 42. And then ApoE3 is somewhere in the middle. So I promised you that after I showed you that, I would narrow it down and summarize it in a more succinct manner. So when you summarize that to what you really need to know, you have this amyloid precursor protein. If it is cleaved by beta gamma secretase, forms A beta 42. That A beta 42, some of it is going to be cleared by ApoEs. What's not cleared is going to start to clump together into polymers, protofibrils, fibrils, and plaques. And these guys have neurotoxicity. They have a lot of different forms of neurotoxicity. We're still finding out ways that A beta 42 kills neurons. We know that it activates NMDA receptors. So those glutamate receptors. We had the NMDA ligand-gated ion channels that bring calcium into the neuron. And when we talked way back about LTP, long-term potentiation, synaptic strengthening, we talked about how synaptic strengthening required calcium to go into a neuron. But if you had too much, then you would get this excitotoxicity. And so A beta 42 can actually independently activate that excitotoxicity. We also know that having the A beta 42 outside of the neurons, just sitting there, is going to cause inflammation and bring in immune cells that are going to further damage the cells, the neurons that are there. We know that if you put A beta 42 in a petri dish with neuronal cells, that it's going to disrupt their cell cycle. And we know that we can do the same thing by putting A beta 42 into petri dishes with healthy cells. Those cells may start to then hyperphosphorylate the tau proteins and create tangles. So there are plenty of ways that A beta 42 can kill neurons. One of the things we're not sure about is how much of a culprit the smaller mono and polymers and protofibrils are versus the larger fibrils and plaques. Some groups say it's really the smaller ones that are causing the most neurotoxicity and that the plaques are really just an after effect. And some groups say that it's the plaques that are the most important as they're bringing in a lot of inflammation into the region. So as far as treating this, our treatment strategies that are under investigation are, of course, things that should keep us from having the neurotoxicity to begin with. So what we'd like to do is block beta secretase to reduce the amount of A beta 42 made. We can just put all that APP into 40. So if we could block beta secretase, that would be great. If we could potentiate our ApoE functions and get rid of A beta 42 better, that would be great. We know that those polymers and plaques in all the different ways are bringing in immune cells that are causing inflammation. So maybe we could try to reduce inflammation. We could try to reduce the microglia, free radicals. We could try to reduce the excitotoxicity that these gave us through NMDA receptors. And we could even try to reduce phosphorylation of the tau proteins or reduce the tangles that are being built up. We have all those things in the works, and they've been in the works for the 17 years I've been teaching this. However, there is a lot of progress in the fact that we do have new drug class that are the anti-A beta 42 antibodies. And as we're going to see, these guys actually come in and bind either to the fibrils and plaques or to the smaller ones, grab onto them, and then inactivate or hold on to them for disposal, like our body just gets rid of antibody antigen complexes. So we had those anti-A beta 42 antibodies. We also have NMDA receptor channel blockers to try to reduce excitotoxicity. And we have reversible acetylcholinesterase inhibitors, which aren't going to help reduce or slow the progression of the disease, but are going to at least boost the cholinergic signaling that we already have, or that we still have. So those A beta 42 monoclonal antibodies are made, here's the antibody here. The antigen itself is that A beta 42. So we're producing antibodies against the A beta 42 antigen. We take this drug, and lecanemab is actually specialized to bind to the smaller forms of it. They're called soluble, and donanemab is specialized to bind to the larger forms when we actually get to fibrils and plaques, and we call those insoluble. And really, that's just based on the size. Think of it like you do soluble fiber and insoluble fiber. You just have them in these smaller, more soluble forms. So I'm not going to have you memorize for me which one binds the soluble and which one binds the insoluble, but I do want you to be able to tell me that as a drug class, these monoclonal antibodies are binding to the A beta 42 externally, and they're removing it. Hopefully by removing it, they can reduce the aggregation, and in the case of donanemab, they may actually even be breaking up some of the plaques. So this is the only drug we have right now that we think actually are slowing the rate of disease progression. Because if the A beta 42 and the plaques are really what is causing the neurodegeneration, then getting rid of these should help slow that degeneration. One of the problems that we have is they do have some pretty significant ADEs. One of the major ADEs that you're going to hear about is due to the fact that epithelial cells for the cerebral blood vessels have enough APP that they can also produce A beta 42 plaques that are going around the blood vessels. So you could have a blood vessel here, and here's your red blood cells, and then the epithelial cells, endothelial, can have plaques surrounding them. So this isn't like arterial plaques like you see with arteriosclerosis. These are those amyloid plaques. If you're going to come in and give an antibody that's going to do great things around the neurons to those plaques, but now you bring it in and it starts attaching to these plaques on the outside of the blood vessels, what's actually happening is in many cases it's interfering with the structure of the blood vessel. And so one of the main ADEs you're going to hear about is leakage of fluid into the brain.